All right. Hello, everyone, and welcome to Noggin Fest 2021. Before we get started, I'd like to give you guys just a little information about Northwest Noggin for those of you who are unfamiliar. Northwest Noggin is a nonprofit neuroscience outreach group that is entirely volunteer driven. And our goal is to bring together students, scientists, educators, and artists to enthuse and inform academic priority K through 12 students, as well as the public about science and art. We hope to spark a lifelong interest in science and art, provide teaching opportunities, and foster understanding and enthusiasm for ongoing scientific research and art in our community. We want to share with you guys some of our favorite moments from our many years of outreach in the greater Portland area and beyond. So please enjoy this quick video of some of this outreach.
This computer is infecting your brain. This computer is infecting your brain. Please deposit all brains in the receptacle next to the computer before you leave. Thank you. This computer is infecting your brain. This computer is infecting your brain. Please deposit all brains in the receptacle next to the computer before you leave. Thank you. This computer is infecting your brain. This computer is infecting your brain. Please deposit all brains in the receptacle next to the computer before you leave. Thank you. Please don't bother to count. The computer will do the counting for you. This computer is making the beats. This computer is making the beats. This computer is keeping you dancing. This computer is keeping me dancing. Dancing disengages your brain. Dancing disengages my brain. This computer is infecting your brain. This computer is infecting my brain. This computer is infecting your brain. This computer is infecting your brain. Please deposit all brains in the receptacle next to the computer before you leave. Thank you. This computer is infecting your brain. This computer is infecting your brain. Please deposit all brains in the receptacle next to the computer before you leave. Thank you. This computer is stealing money from the arts. This computer is eroding information credibility. This computer tracks your movements. This computer knows your spending habits. This computer remembers everything that you do. This computer will change the way you think. This computer is making the beats. This computer is keeping you dancing. Dancing disengages the brain. This computer is infecting your 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 brain. Do not try to count. The computer will do the counting for you. This computer will replace your brain. This computer will replace your brain. Please deposit all brains in the receptacle next to the computer before you leave. Thank you. Please deposit all brains in the receptacle next to the computer before you leave. Thank you. Please deposit all brains in the receptacle next to the computer before you leave. Thank you. Please deposit all brains in the receptacle next to the computer before you go. Thank you. Please deposit all brains in the receptacle next to the computer before you leave. Thank you.
All right. So now that I've introduced Northwest Noggin, let's get on with Noggin Fest. We are so happy to have all of you guys here tonight to join in our annual celebration of neuroscience, art, and music. Tonight we will be hearing some exciting research in neuroscience related to monkey brains, the gut-brain axis, hearing, and psychedelics. We also have a handful of talented musicians here with us to share their music. Additionally, throughout the event, we'll be hosting a live art auction featuring plenty of stunning neuro-related art pieces. So please click on the link in the chat to check out our auction page. All proceeds from the auction will go towards supporting our artists, as well as towards supporting, sponsoring our students from Portland State University who will be presenting at the Society for Neuroscience in Chicago this fall. We are also selling raffle tickets at $5 a piece for a bunch of really cool prizes. You might notice that some of them are here behind me. We've got all kinds of fun 3D printed brains and a bunch of other goodies as well. So go ahead and click on the link in the chat to check out some of those prizes, and make sure to buy your tickets before the sale ends tonight at 8 o'clock. And we would like to give a huge thank you to our sponsors, the Dana Foundation's Brain Awareness Week program and On Point Community Credit Union. We'd also like to thank Fort George Beer, Paxton Gate, and Ear Trumpet Labs for their generous donations to our raffle prizes and art auction. And lastly, I'd like to give one more big thank you to all of our artists, presenters, musicians, and lovely volunteers who have put in a lot of work to make this event possible. To kick off Noggin Fest, I'd like to introduce Gail Stonebarger with a land acknowledgement.
Hi, everyone. Like Carly said, I'm just going to lead us in a land acknowledgement. I'd like to start by pointing out I am currently in Portland, Oregon, which is unceded land of multiple tribes, including the Multnomah tribes, Clackamas tribes, and then a group of about 30 tribes called the Confederation of the Grand Ronde. In the chat, you'll see we actually have a link to nativeland.ca, which is one of my favorite resources. I'd encourage all of you to click that link, type in your address of where you live and work, and see whose traditional land you're on currently. Anyone is welcome to share that in the chat, or you can just let it be known and appreciate that we are on land that was never actually given to us. So like I said, links in the chat, I'd encourage you to do that. And I will turn it back over to Carly to introduce our first artist.
Thank you, Gail, for that land acknowledgement. Here in just a moment, we're going to welcome Fish Martinez, aka 28 the Native, who will be performing with us, or for us, I should say. Fish is Siletz, Shasta, Tutuni, Yurok, Mescalero, Apache, Azteca, and German. And he is an educator and a member of the Hip Hop Congress. And he'd like to share his music with you all today. So give us just a moment while we get him all set up.
All right, let's have some fun. Yeah, check it. Okay. Embracively creative, going by any name that fits. Perfect at the time or right after you hear the chimes. Pacing myself at a full sprint. Native out competing, ciphers never miss. Leading all about the healing way. Always look eye, very inspired lines like family ties. What an Alex P. Keaton design. If Alex P. Keaton design was screw the government like straight 9. Restoration by any means, collabing minds gets that new hit. Recipe slow cooked, so it's so good. Not even too good. Sour tasting like some yogurt with live culture. My entire life has passed right by, so I'm flipping formidable syllables, healing by touching the patient with doctorly persuasion. I blaze in amazement, making you wonder where the days went. While this Northwest resident, captivating my elevating situations. I'm bread breaking and crumbs manifest. That's why I feed with the best. The spice mixing beats with this. It's tribally influence. I ain't a veil, Julian. CED, with sportsmanship on the indigenous tip. Rips as if my name was Hannah. Shout outs to Emil Anthony, this how we do it, all about the family. My mind states encoded, genetics devoted, so hopeless. Yo, what? I can't lose focus. Dreaming, let's go. I'm dreaming. Oh, I got this dream, been walking into the existence of being DGS and on the scene, tribally influencing. That's what the message be. Recognize the C-U-L-G-U-R-E. That's what I'm breathing. Yeah, you heard me right, and now you've heard me right. Inscribe these scribbles straight from the middle of my spiritual outwardly. Pass it over here. If you so choose, cause I'm a soul choose, my soul's bruised, trying to get back on that path. Quick to learn, still get scars from the burns. Took that turn for the worse, don't take too long to do with this verse. Just happy to be alive, all about the elements of vibe. Four directions and triplicate with my brights. Gets you listening, healing with this medicine. You can put it on the ones and them twos. Beat talks with the cuts like what? LP gets me dreaming. Yeah. These lyrics in case, just in case. I'm dreaming, and I'm very still in that dream state, wide awake. Now that I've got a taste, rapid eye movements, driven to create, not trying to let the energy go to waste. My mind states, spinning wheels, rap race, knowing that this water might be laced with strychnine. While we still have a chance, one big team, extended family, from here to Germany, unite peacefully, visions to reality. Got me traveling, scribbling, capturing, setting goals and accomplishing. Giving thanks, gotta buy more than star visuals by my family side. Cascaded, braided, spotlights, warrior imagery exposed by the drones. Bandanas laced across the face, unity is strength. 7 generations all up in the place, native pride with the faith. from the little sage. Bless up the space while I rock the stage. Golden aerosols, I'ma act my age. To grow my braids with that length. BGSing on the scene. Tribally influencing, that's what the message be. Recognize the C-U-L-T-U-R-E. Yeah, I got this dream. Yeah. Okay. It's that cold hard rocker, street entertainer, going for the zone just to feel the rush as my braids brush past the mic. Can a nectar 503 in the sector with my liquid tree. Concentrate, bounce that energy till it gets connected. Tell them that I said it's headed for prophetic. I know you won't forget it, embedded in my DNA like chromosomes. Let it be known, still that surrogate father from microphones. Macro with the flow, no doubt. Or without, I came to compete. Rock, CL, Snoop, just watch me. It's all in the mix, my talent exists. You can raise up your fists. I was raised up with this. Mental over everything, so you can underdig, relate with you ain't spits. Hojo, gonna keys. To all human beings, PGS, weapon collective, no ceiling, cause we used to see it. Clouds in the sky, especially when the rain drops. Northwest, I don't need the name drop. Got more fire in this ring than Mount Doom. Mystical, mentally, Dr. Doom. You might presume, hell yeah, epic ever since the womb. Mad love to my Madre Bonnie for all she's endured. So excuse me if I'm rocking hip hop and I think you know the name. But the trick's not my game, doesn't mean I haven't lied. Now I try to do what's right, write and recite on sight, vision precise, slice obsidian, urban city. Oh yeah, I know you was waiting for me to say Indian, but I'm not. Just cause Columbus who was lost, stop. But I'm not full of hate. Oh, we can kick it like this always. When Rec Rockers music is on play. Yeah, you can find me on stage. Passionate to entertain. Water is life. Water is life. It's not to be sold, a blessing to behold. Water is life. Heal the abuse from the elders to the youth. Water is life. Protect our access and waterways. Drip, drop, and evaporate. Splish, splash, now I'm wide awake. You can crush it or slush it. Compact and attack with an icy glass solid state. Or wait for the snowflake. Beautify the landscape as it falls, erodes and shapes. Sip or drink, boil with, make medicine with, dilute or cleanse. Spend the weekend in it with friends. See straight through and feel the force. Typhoon or riptides, recreate or seek to survive worldwide. I love out the proper alkaline, redirect or divide. Yo, respect the flow. Stop that drastic pesticidal leaking plastic containment. No more fluoridation. Sweat it out. Know the heathen routes. Purify. Give thanks for the blessings. Life cycle, so we recycle. Water spouts and bubbles up. Drops knowledge from the clouds. Learn the lessons, no doubt. Good looking out. Protectors, not protesters. Passionate activists, warriors in the game. It's what you claim. All about the front lines without the fame. Pairs up, chill out, and hold it down. From yourself to the universe. Truth hurts, monopoly properties. Water works. Conscious as the water drop, I'm aligned with a moto's mind through the power of prayer. Bringing molecular change over time. Water crystals don't have to stay polluted. United, that's how we do it. With a hashtag, no doubt. For example, corrupt. Stop raping our land, what's up? $1 billion schemes, manipulate, manifest destinations to control, dig holes, then act like they don't mind. Illusionists with a twist, put them on the terrorist list. About to head to North Dakota to up the quota, pull over. Gotta taste some of that Nestle freeze straight from the spring. Stop any government that tries to lie, separate and divide. Water is life. Water is life. It's not to be sold, a blessing to behold. Water is life. Heal the abuse from the elders to the youth. Water is life. Protect our access in waterways. Shush. Whew. Thank you very much.
Thank you, Fish, for that awesome and lively performance. It was a lot of fun. Next up, we have Gail Stonebarger coming back on screen, who is pursuing graduate studies at OHSU and will be discussing her research with some of the oldest monkey brains in the world. And just a heads up for our audience members, there will be a Q&A session at the end of every research presentation. So be sure to hang on to your questions if you have any. And for each Q&A session following the presentation, you will all have the opportunity to vote for your favorite questions. And those ones with the highest votes will be prioritized most by our speakers. So without further ado, I welcome Gail back to share her research with us.
Hi, everybody. Let me get my screen shared. All right. So like Carly said, my name is Gail Stonebarger, and I am hopefully finishing up a PhD program in behavioral neuroscience at Oregon Health and Science University. And I work at the Oregon National Primate Research Center, which is a campus of OHSU that's out in Hillsborough. And as it sounds, I work with monkeys. So I'm going to talk about why it is that we study the oldest monkeys in the world. So initially, it's really important for us to know that the human population as a whole is getting older. So we all know that average lifespan back, hundreds and hundreds of years ago was 30 to 40 years old. And as medicine has increased and become modernized, we are now able to keep people alive for much longer. So on this chart, we can just see that the number of people that are over age 65 is rapidly increasing. By 2050. It's supposed to be almost 90 million people in the US. And then those over the age of 85 are also rapidly increasing. It's not as steep of a slope for those of us who are into statistics, but if we look at the numbers from 2010 to 2050, those age 65 and older are going to about double, and then it's almost supposed to quadruple for those who are over 85. So since so much of the population is actually getting older and staying alive for longer, it's really important for us to be able to study what's happening to the brain, not only as we develop and go through life, but also as we start these processes of overaging. So like I said, study monkeys. I study the rhesus macaque. And the reason that we use these is that they're a more clinically translational model for human aging. So you can imagine that rats and mice are a really good baseline to start out with. But if we look at things like genetic relations, monkeys obviously are evolutionarily much closer to humans. And that means that about 93% of all of our genetics are shared with rhesus monkeys. And then partially due to that, rhesus macaques actually have a highly developed neocortex, which is different from what rodents have. So monkeys have most of the same, both cortical and subcortical brain regions that humans do. And they're in pretty much the same places. It's just sort of a condensed version of that brain, as monkeys are significantly smaller. We also, with monkeys, see that they have these similar life stages to humans. Mice and rats, if you're trying to study development, you only have a couple days window, and similar with aging, right? This period of transition into old age is much shorter when we look at rodent models because their lifespan is so short. It's usually about 2 years. Social structures are another thing that we can look at. So there are social hierarchies with mice and rats and other laboratory animals, but monkeys are, especially macaques, have very strict hierarchies, and their social interactions are actually very similar to humans. So for instance, I have multiple colleagues who study alcohol intake and alcohol addiction in monkeys, and we see these similar habits where certain monkeys consume a lot more alcohol voluntarily when they are around their peers and when they have access to their friends, basically. And certain monkeys drink a lot more when they're in isolation. So this is something people are looking at in terms of social behaviors, and we don't see the same type of social patterning in rodents. The final thing that I like to point out is that monkeys are capable of much more complex cognitive tasks than rodents are. We can put rodents in mazes, but we can actually teach the monkeys at the Primate Center how to play games on iPads. So a lot of the testing that we do with cognition and dementia in the monkey model is actually basically an iPad version of what neurologists are giving their human patients to determine what type of cognitive either cognitive disabilities or cognitive, whether they're staying cognitively healthy across age. And so we basically can translate that directly into a monkey test, which is really helpful when we're looking at the way monkeys and humans age. So there's an extra bullet point on here, I apologize. But just the monkeys that I study specifically came from the National Institute on Aging, which is a subsection of the National Institutes of Health. And they were actually in this longitudinal lifespan aging study. So they were on specific diets. They weren't allowed to become obese. And the principal investigators wanted to see whether that would extend their lifespan. What happened is the monkeys that were on a calorically restricted diet actually didn't live any longer than their peers, but what we call their health span was significantly longer. So they went multiple years into aging without getting these aging related diseases. Endometriosis is a really common one, diabetes, things like that, that primates, including humans, are more likely to get as they age. So the monkeys that ate fewer calories were actually able to stay healthy for longer, which in my opinion is actually more important for human health. Personally, I would rather live a happier life for a shorter amount of time than live longer with more of these diseases. So when we look at these monkey brains, I didn't mention, but it was on the slide, that monkeys age at about 1 to 3 of what humans age, or 3 to 1, I should say. And so the very oldest monkeys we have are equivalent to the very oldest humans ever recorded in existence. So 44 is the oldest animal, and that would be nearly 135 in human years. So we're looking at these brains to see whether these animals look like they have neuropathology, or whether they look closer to humans who don't actually get this dementia as they age. So at first glance, this is from a paper that my lab published last year. And these are all different phenotypes of what amyloid beta is. Amyloid beta is the protein that makes up the plaques half of plaques and tangles. When you hear about the neuropathology of Alzheimer's disease, if you know much about it besides just the dementia, the first things you start hearing about are these plaques and tangles. So these plaques are made of amyloid beta, and they form these different phenotypes. They form all these different clusters outside of cells and then also in the vasculature. And it's thought that that is impeding some of the functioning in both Alzheimer's and healthy brains. It's really important for us to remember, though, that even though we know and think about these plaques as sort of an Alzheimer's symptom, they actually start to occur in late middle age in all humans. So there's really very, very little chance that someone would die naturally or in old age and have no plaques. we just see this really elevated amount with Alzheimer's pathology. And what I found in the paper that I recently published is that, yes, amyloid beta plaques accumulate across age in these monkeys, but even the very, very oldest monkeys didn't have the same number of plaques that any humans who are diagnosed with Alzheimer's disease would have. So that's one of the theories of why monkeys don't get the same overt dementia that humans do, is that their amyloid beta just actually doesn't reach high enough levels to be pathological. And then the second half of this, we have the plaques and the tangles, is the tangles half, obviously. So phosphorylated tau, or p-tau for short, is the tangles aspect. And this actually starts intracellularly. So it's this protein that begins to accumulate inside cells. And these 4 images are just to show you what human and monkey pathology looks like and how similar it is. So this is the same way of staining for the same type of protein. And on the left side, we see human images. So this is in the hippocampus. Anything that's stained is this phosphorylated tau. And so we see it kind of zoomed out in the hippocampal CA regions. And then we also can see this closer objective of what the proteins look like inside the neurons. So those dark blobs are actually neurons that are being taken over by this protein that impedes cell functioning. And then on the right side, we see basically the same views in the same brain regions of what that looks like in the rhesus macaque. These are really old monkeys. This animal actually lived longer than a significant number of monkeys do. It was well into its 30s. So that's another thing that we need to keep in mind, that these levels start to accumulate. It looks like past when these monkeys would actually normally be living. So if we put all of this together, it's been proposed that monkeys are a good model for Alzheimer's disease, because unlike rodents, they do naturally accumulate these proteins. So if we look in rodent models at Alzheimer's disease, we actually have to induce the accumulation of these proteins, which means that we can't look at how they start and how they spread in the same way that we can in a monkey, where it's already happening naturally. But if we look at sort of the key components of Alzheimer's disease, we have this overt dementia, which any of you who have known anyone with Alzheimer's disease will understand that it's this very fast decline in cognitive processing that monkeys just don't get. They naturally, their cognitive processing skills decline with age in the same way that humans do, but not in the way that humans with neurodegenerative diseases do. They also don't reach this critical level of amyloid beta in their brains, even the very, very oldest monkeys to ever exist. And then what I haven't talked about yet is that this tau, this phosphorylated tau in humans, depending on what neuropathological disease they have, there are different patterns of spread. And I have worked with a clinical neuropathologist for quite a while on this, and we can't find a single disease of aging in humans that actually has the same type of spread, right? So tau starts in the same place and then continues to spread to brain areas in the same order. So after the last 4 years of research, we kind of came to the conclusion that as much as we want it to be a model of Alzheimer's disease, we actually can't call these monkeys a model for Alzheimer's disease because they don't reach these benchmarks of pathology. So I actually just want to pose to you guys, if anyone's comfortable either unmuting or putting in the chat, what from this we can actually get from studying these monkeys if we thought they were going to be a model for Alzheimer's and they're now not any longer. I've taught a couple classes on Zoom recently, so I can wait a couple minutes if anyone has ideas. But if anyone has ideas, I'd love to hear what you think. I hear someone, it might be Bill. All right, you guys are shy, that's fine. I'll just give you the answer. So my dissertation is currently is actually looking at basically why rhesus macaques don't get Alzheimer's, even though they have sort of all the same baseline things that could lead to it as humans do. So monkeys have, these plaques and these tangles, and they live in some cases longer relatively than a lot of humans do, but they're still not getting this overt pathology. So there are lots of sort of theories on why this would be. One of the big ones that would seem to be obvious is that monkeys, don't reach this level of clinical pathology, like I was saying. And so that might be environmentally driven. One of my favorite facts actually about that is that pet dogs are one of the only animals that also develops, outside of humans, this Alzheimer's phenotype. So they get this intense decline, they get plaques, and there's evidence of tangles. And that kind of points at a possibly environmental aspect of this. Because if you think about it, pet dogs are really one of the only other species that is exposed to pretty much all of the same things that humans are, which is really interesting to me, that it would be possibly something outside of just our natural chemistry that could be causing this. So I think I've gone a little bit long, but I just want to make sure and acknowledge everyone, not everyone, a lot of the people who have helped me. On the side, this is my dog, Moose. Grad school is really, really hard, and Moose has been just such a great pup to have around and go on hikes with, as you can see. But mainly I want to thank my PIs, Dr. Henrik Urbanski, who is who I work with. He's my mentor at the Primate Center. And then Steve Kohama, who's a staff scientist and a mentor. Steve Dash and Opal Steyer Wilburn are also in the lab and have helped me with some of this data. Julie Madison and Kelly Vaughn work at the National Institute on Aging. Doug Rosina and Sam Calderazzo are at Boston University, and they've also been integrally involved in the experimental design aspects of these. The clinical neuropathologist at OHSU has helped me a lot. I belong to both behavioral neuroscience at OHSU and the Division of Neuroscience at the Primate Research Center. And they've both supported me throughout my career. To thank my funding, the students who have helped me get through this. I always want to make sure to thank all of the monkeys that have been used in my study, and also in all of the studies that have led up to the point where I get to study them. Of course, Bill and all of the Northwest Noggin volunteers who made this possible. This is such a cool event, and I'm really glad that you guys figured out how to make it be online this year. And then also just thank you to everyone who's in the audience right now. I appreciate you taking the time to come learn about some neuroscience and art. So that's all I have. And if anyone has questions, I can go ahead and answer.
We have a number of questions here, actually. And Gail, thank you very, very much for that talk, and also for the land acknowledgement as well. And let me just pull up the chat here, because we had quite a number. So the first one here is, does using iPads actually better model human behavior, since so many humans are now completely tied to their own screens? Like
I would say yes. Yeah, yes, like tonight. I would say yes, because it is a lot of rodent studies, if you think about it. They're going in mazes and they're doing very simple but movement-based cognition. And it is a different type of task if you're sitting in front of your iPad and you're using your spatial reasoning on this screen, as opposed to the world around you, which is what most of us do. And like I said, it's also 3 minutes, cool. It's also has to do with the type of tests that we can give. So we can't give mice very many, complicated tasks, whereas monkeys, we can, if we motivate them with raisins or M&Ms or Cheerios, those tend to be the favorites. They'll learn really intense tasks. Like most things that you could teach a kid or an adolescent on an iPad, they can learn how to do. So it's really cool.
Okay, we have another question here. What would a calorically restricted diet look like for people? Would it be easy for many people?
No. So these monkeys that were calorically restricted were restricted by 30%. So they ate 70% of the calories that, based on like standards they would be consuming for their weight and sex. So it would be, take what you are supposed to eat based on the nutritional regulations and then cut that down by 30%. And that would be what the calorie restriction is.
So you may have answered this. There's a question here about like, are there any additional animals that are a better model for Alzheimer's disease? It sounds like maybe dogs, right?
Yeah, so dogs were not quite sure on the different phosphorylation sites of the tau, which is like an intense way of saying that we don't know whether the tau protein is the same in dogs as it is in humans. But if we look in monkeys, it is actually the same phosphorylation sites. We use the same chemicals to detect the proteins as clinicians do in the lab when people die to look at human brains. So there are lots of primates that sort of have both plaques and tangles, but the tangles in rhesus macaques actually are, rhesus macaques and higher primates are the ones that are closest to humans. So.
Okay, so we have, I'm not sure how much time left, but there's another question here. Would the monkeys possibly develop Alzheimer's disease if they had more stress and less sleep?
So that's one thing I'm looking at actually, is looking at brains of monkeys that were on high fat diets. I haven't gotten too much into this data, but the Primate Center has a big population of monkeys that are kept on American style diets. So they're high fat and high sugar, and they mimic what most Americans eat. And we're looking at those in terms of whether multiple sort of negative things will build up to an Alzheimer's-like phenotype. So some of them also are overreactomized and not given estrogen back. So that's another thing. It's like being postmenopausal, and that's also bad for your brain in some cases. So I'm out of time, and so I'm going to stop talking, but thank you guys for being engaged and for your questions, and feel free to in the chat or something. If you have other questions, you can email me, whatever works. So thank you, guys.
Thank you.
Yeah, thank you so much, Gail. All right, next up, we are very excited to introduce to you all a local Portland band named Stella Ruse.
There we go. All right, hi everyone. As Carly mentioned, we are a Portland band, and we're gonna play you a few songs that have not been released yet, but we're working on recording currently for our current album that's coming up, and that'll be available on Spotify and Apple Music and all those places, hopefully towards the end of this year. We're working on recording it, but you know how recording works when you can't actually have access to a studio due to COVID and all that stuff. So we're just trying to go for a home recording route, and hopefully that'll work out and sound awesome. Let's see, where's my pick? Here we go. All right, you ready, Josh? Yep. Make sure my guitar works. All right, the first song we're gonna play for you guys is called Moving the Fulcrum Over. Here we go. Sky.
deals I've held since I was young. I know there's more to you than leaving. It just shows we're green enough to look the other way and settle down. Stop freaking out. No, I've slowed down since I saw you last. No looking back. In every dream I have, I'm running. The ocean line is up in flames and I'm alone. The clothes I wear feel unbecoming of me. Feels like everything I've said's been said before by a man more deserving of my lauded life, a half-assed life. Old ideas of love, these bold ideas of love, these sad ideas come pouring out, but I've since learned to sit around for love. Cool. Thank you.
Alrighty. I'll trade you these headphones, or I think we were keeping the same guitars, right? You were just gonna bring the mic over. Here, you can have the headphones. Cool. Sorry. They were saying our volumes were low for a bit. I think it sounded okay in the headphones, so hopefully we're all good.
Cool, cool. So you have to play louder, right?
Something like that.
All right, this second song does not have a name,
Nice.
but it is my song. So we're gonna play it. Are you ready, Jake?
Yep.
Always try to help you out. Your back is turned towards the crowd. The guy up front doesn't know a thing about how his brain works, and he stumbles around. far too windy and bendy to settle down right here. And I hope soon to love what I've had and move on from my old memories. Yeah, I'll let go and forget how to act, but that stranger will never come home. And it was worth it all to know that your kindness has withered your bones. Awesome. Thank you very much. Those are our 2 songs we had for you today.
Fantastic. Thank you guys so much for sharing with us. Again, that was Stella Ruse, and I think I heard them say that they have an album coming out sometime maybe later this year, so you guys will have to keep an eye out for that. So right now we're going to go into a quick 5-minute break for everybody, during which we will be showcasing some of the art that was donated to our art auction, so you can look at that. Now is a good time to also buy some raffle tickets, check out the art auction page, and run to the restroom real fast. We'll be starting back up with some more fascinating research in about 5 minutes, around 7:05. We'll see you then. All right, and welcome back, everybody. So as people start returning to their seats, I would like to begin to introduce our next speaker, Dr. Heather Zwicky, who is a well-loved professor at the National University of Natural Medicine here in Portland, Oregon. She will be sharing with us some of her research on the gut-brain axis. So Heather, come on over.
There's the volume. Hi, everyone. I am Heather, and I am going to talk to you today about gut-brain axis. So, what is an immunologist doing talking at Noggin Fest? Well, for about the last 15 years, I have been studying neuroinflammation. And you might think that neuroinflammation starts in the brain, but actually, one of the things we have learned is that neuroinflammation starts in the gut. So, what is the gut-brain connection? Well, about 15 years ago, we started looking at things like how come we could give somebody a probiotic and it actually had an effect on their depression? What was going on there? And initially people thought, well, this must be psychosomatic. It's all placebo effect. But it turns out it wasn't placebo effect. And so people started looking at what the gut-brain connection was. Now you have to remember that historically, Descartes separated all the different organ systems. So we were studying the brain separately from studying everything else. And when we started doing that, we stopped remembering that the brain and the gut are connected. And it turns out that there's not only a physical connection, the vagus nerve, but there's also a chemical connection. And that chemical connection is created by our microbiome. Now, I'm sure many of you are familiar with the microbiome because it's a popular topic these days. But in case you aren't, let's just get caught up, okay? So we start with microbiota. And microbiota are the assortment of microbes that live in our gut, on our skin, in our lungs. All these places that we previously didn't even realize there were microbes, we're now starting to find out that there are microbes there. Places we thought were sterile, turns out there's microbes. And what's interesting about these microbes is that they're producing all sorts of factors that can interact with our brain. So even though something is being produced in the gut, it can influence the brain. Now, I want to catch you up in the way that we think about the microbiota, because a lot has changed just within the last couple of years. It used to be that we thought if your gut microbes were out of balance, we would call that dysbiosis. And dysbiosis meant that you had more bad microbes in your gut than you had good microbes. Now we're starting to think about that in a different way. We're starting to think about the gut microbiome as an ecosystem. To think about ecosystems, I think it's helpful to use a model. And the wolves in Yellowstone are a great model of an ecosystem. So you may be aware that in the 1920s, gray wolves were removed from Yellowstone. And as a result of that, a lot of other species of animals started growing, and they started growing in rapid numbers. And because they were growing, let's take beavers as a good example. If there's more beavers, what do you think is going to happen? Well, more trees are going to get cut down, right? And when more trees are cut down, then we actually see that more rivers are going to change the way that they flow. And if rivers are changing the way that they flow, that might change the fish that are in the rivers, and it might change the amphibians that are in the rivers. Then in 1995, we added the gray wolf back to Yellowstone. And the gray wolf came in and it started killing off some of the elk herds. It started killing off some of the coyotes and some of the beavers. And what we saw is that even the direction of the rivers changed when we added the gray wolf. Now, here's something that's important about that, because we didn't add hundreds and hundreds of gray wolves. What we did is we added 5, and they reproduced, and now there's 50. And 50 gray wolves can completely change the way that the rivers flow. That is because the gray wolf is what is called a keystone species. A keystone species is something you don't need to have a whole lot of, but it has a huge impact on the environment around it. And that turns out to be really important for our microbiome. Because, as it turns out, our microbiome is like an ecosystem, and different people have different ecosystems. Now, if you think about ecosystems, you can think about a forest or a desert or marine or grassland. They're all different, right? But none of them are healthier or unhealthier than the others. So the problem when we think about ecosystems is that we've been treating everybody pretty much the same, but you might have a forest ecosystem and I might have a marine ecosystem. And what we need for our different ecosystems may actually differ. So what I want you to first remember about the microbiome is that it is not about the numbers of microbes that are there. It's about what they do. It's what the microbes do. So let's think about this a little bit. What happens? You eat food, right? The food feeds the microbes in your gut, on your skin, in your lungs, etc. And then the microbes make things that go into your blood and go to the brain. Now, in case you haven't caught up because you're not immersed in this field like I am, prebiotics are food. These are things like onions, Jerusalem artichoke, mushrooms, chicory. They're food that have fibers that are hard to digest, and these feed your microbes. Probiotics are bacteria, but most, if not all, the bacteria in a bottle of probiotics are dead. So really what we think is it's probably the metabolites, the essentially microbe poop that is feeding your microbiome. Postbiotics are metabolites from bacteria, and usually we harvest these metabolites from poop. Parabiotics are heat-killed probiotics. So if we take our probiotics bottle and we throw it in the oven, when those probiotics die, they produce all sorts of different factors, and it turns out those things are healthy. So those heat-killed probiotics are parabiotics. And the research from this one is now going to be coming out of Belgium. And then synbiotics. And synbiotics is the combination of prebiotics plus probiotics. So when we feed a prebiotic to a probiotic, we call it a synbiotic. Here's why this is important. It turns out that what you need for your microbiome is diversity. What you're looking at here is what's called alpha diversity. And what we want is right over here on the right. We want high levels of alpha diversity. We can eat any kind of diet, omnivore, vegetarian, omnivore, no red meat, vegetarian that eats seafood, vegan. All of those colors are over here on the right-hand side. What matters most is that you consume more than 30 types of plants per week. So this is a study done by the American Gut Project at UCSD. And what they showed is that 30 types of plants per week was the most important thing. So 30 types of plants per week. So what I'm going to tell you to do is get your 30. How are you going to get your 30? You're going to try to eat 30 different types of plants per week in order to have the healthiest microbiome and the healthiest brain that you can have. Because if we feed our microbiome makes metabolites. Now, what are these metabolites? Well, some of them are short chain fatty acids and some are bile acids. They actually deconjugate bile acids. But microbiome is also responsible for making neurotransmitters. And here's one of the relationships to the brain. So the short chain fatty acids are something I'm interested in as an immunologist, but it turns out neurotransmitters also have an effect on the immune system, as do vitamins and some of the other peptides. And the neurotransmitters that the microbes make include dopamine. In fact, your microbiome is making somewhere between 50 and 60% of your dopamine. It makes almost 95% of your serotonin, and it makes a large percentage of GABA. We're not 100% sure of the amount of GABA that your microbiome is making. Let me say that again. Your microbes in your gut are making your neurotransmitters. Who knew? Who knew? So everything you eat impacts the neurotransmitters that you make. Now, this is super important because remember, dopamine is our reward neurotransmitter. This is how we feel positive reward. Like when you find a $20 bill in your pocket and you go, woohoo, that's dopamine. Serotonin is involved in happiness, and GABA lets us sleep through the night. This is our relaxation neurotransmitter. So if you're one of those people who wakes up in the middle of the night, you got to work on your GABA. What does that mean? You got to work on your diet. So I got into this story because of Parkinson's. In 2015, there was an article that was published that showed that people with Parkinson's were missing certain species of microbe in their gut. And when they're missing that certain species of microbe in their gut, it turns out they can't walk. So the microbe that was demonstrated to be missing was called Prevotella. And when Prevotella was gone, then other microbes filled in. And as it turned out, it was affecting gait difficulty in Parkinson's. We always thought the gait difficulty was in the brain. It turns out it wasn't. It was in the gut. I'm going to show you some recent data that shows that this is the case. So this is characterizing dysbiosis of gut microbiome in Parkinson's disease. And this is a paper that just came out this year. At the bottom, you've got the different species of microbes. And I realize you can't really read those. Doesn't matter. They're not that important. What matters is that our cases of Parkinson's are in blue, and our people who don't have Parkinson's are in orange. And what we can see is that there are 3 different species of microbe here that are overgrown in Parkinson's, and there are 10 species of microbe that are missing or undergrown. These guys are almost missing. These guys are undergrown up here. And then there's these other 2 species that we'll talk about in just a second. But these 10 species of microbes that are undergrown are the short chain fatty acid producers. They're also the microbes that produce dopamine. So people with Parkinson's missing dopamine, part of it is definitely the substantia nigra in the brain, but the other part of it is their gut microbes. And if we don't fix this one, it doesn't matter what we do with the brain. Now, these other 2 species happen to be lactobacillus and bifidobacterium, and those are the 2 bacterium that we put in our probiotics. So hold on, look, they're overgrown in Parkinson's. They're not undergrown. So that's kind of a challenge here because it says to us, should we be giving probiotics to people with Parkinson's? It turns out that when you give people with Parkinson's L-DOPA, it causes the overgrowth of these bacteria. The fact that the gut and the brain are so intimately related means that if you want to hack the gut-brain connection, you can do it from either organ. You can either come at it from the brain side, maybe using psychedelics or nootropics, or the mind-body connection, so meditation or breathing, or you can do it from the gut side. You can use prebiotics, probiotics, postbiotics, parabiotics, symbiotics, food, and fasting. And I was really interested to see the last speaker talk about fasting because we know that fasting also has an impact on the gut microbiota and it decreases inflammation. And then vitamins and omega-3s as well. So I want to thank all of the Noggin Fest organizers and participants. I also want to do an Indigenous land acknowledgement and acknowledge not only the place where I am at NUNM, but also Klatskanai, where I live. And if you want to reach me, you can follow me on Instagram or you can go to my website. I'm happy to take any questions.
Thank you very, very much. And that definitely has a lot of us, I think, thinking about, dietary changes. And there are a number of questions here that relate directly to that. So one is, how much do organic foods benefit the gut-brain axis in comparison to non-organic foods, if at all?
Absolutely, organic foods are amazingly beneficial. And the reason is that foods that have pesticides kill gut microbiota. So there's increasing data. In Parkinson's, we know that 70% of Parkinson's is environmental. And we know that it's because we've killed off these different species of microbes. And the number 1 killer of microbes are pesticides. Number 2, things like BPAs, air pollution, antibiotics, actually other kinds of medications that aren't antibiotics. So we need to keep our gut microbes healthy and happy, and pesticides don't do that.
Wow. So another question is, are dietary changes actually now considered like a medical intervention for mental health disorders specifically, because of the clear impact of the microbiome on the brain?
It's a great question. And we would think that that would be really obvious. But one of the things that we're discovering is that most physicians, when they go to medical school, get roughly 3 to 4 hours of nutrition in their entire medical school career. So usually you aren't going to get a whole lot of nutrition advice from a physician. You're actually going to have to go to a naturopathic physician or a nutritionist to get some medical advice. If you go to those folks, absolutely, you'll find that you can, Hippocrates said it, let food be thy medicine, right? So we've known it for a long time. We just don't necessarily follow the advice.
Wow. So here's another question. Do you think that our overly sanitized environment could have a negative effect on our brains, especially with so much hand sanitizer and all the precautions we've been taking for COVID?
Absolutely. Absolutely. One of the things we're seeing is that people are totally wiping out their skin microbiome with hand sanitizer. And so we're seeing a whole lot of rashes come in because the skin microbiome is just gone. And wearing masks. Remember that when we wear masks, we're no longer exposed to normal microbiota in our environment. And so we're seeing a lot more allergies and asthma appear as a result. So hopefully we can get the vaccines rolled out so that we can revive our microbiome without masks.
So we had another question. Can you explain more about how neurotransmitters are produced in the gut? They were very struck by that.
Well, so there's two ways. The first way is that the microbiota themselves actually produce the chemical serotonin. But the second is that they can stimulate enterocytes in the gut, so cells in the gut, to produce neurotransmitters. So there's two different ways that the neurotransmitters are being made in the gut. What most people don't know is that neurotransmitters are responsible for you pooping, right? So the contraction of the colon, that's coming from dopamine. Dopamine contracts the colon. If you don't have dopamine, you're going to be constipated. And serotonin is regulating how poop moves down your intestine. So again, you don't have serotonin, you've got a problem on your hands. And GABA, if you think about pooping, you've got to squeeze some stuff through a little tiny hole. GABA actually numbs that spot so that you don't feel pain when you poop. So if you're feeling pain when you poop, that means that you don't have enough GABA.
That's really interesting. So is food a better source of probiotics than supplements? Was another question.
Yes, food is live food, things like kombucha and sauerkraut, absolutely a better source of probiotics or live microbes than supplements. But also keep in mind that we want diversity, right? So people who think that we should take the same probiotic every day or eat the exact same kombucha every day, that's not what your microbes want. They want as much diversity as possible. So trade it up with your friends. Everybody buy a different bottle of probiotics and then trade it up so that you have different probiotics that you're exposed to, or trade up foods, because that's definitely what's better for your microbiome.
Another question is it true that Americans tend to have far less diverse microbiomes than people from other parts of the world?
The only ones who are less diverse than us is the UK. Turns out bangers and mash don't give you a whole lot of microbes. So yeah, we're pretty bad. We're like 30-fold less than most other countries. Yeah, it's pretty bad. We eat a lot less vegetables. I mean, come on. In the United States, most people count ketchup as a vegetable. It's not.
So how does drinking lots of coffee all day long impact the gut biome?
Well, I imagine that you're going to pee and poop a lot more because you're speeding up all those systems. But I don't know exactly which microbes are going to change.
Well, on that note, I think we're going to finish up. And thank you very much, Dr. Zwicky, for joining us tonight. We really appreciate it.
You betcha. Have a great Noggin Fest.
Cheers.
All right, and I'll just second that appreciation for Dr. Zwicky. Thank you so much. We were so thrilled to have you present your research tonight. I think you've probably inspired a lot of people to go eat some more vegetables, so that's always a good thing. Next up, we are also very excited to hear from Dr. Theanne Griffith, who is an esteemed neuroscientist and children's book author. And she's going to be reading us one of her books, as well as conducting a brain bake activity that she's created to go along with her books. So without further ado, here is Theanne Griffith.
Hello, everybody. I'm so happy to be here. Thank you for the invitation, Noggin Fest crew. So my name again is Theanne Griffith, and I'm a neuroscientist at the University of California, Davis, as well as a children's book author. So today I'm going to be reading from the second book in my series. The series is called The Magnificent Makers, and this book is called Brain trouble, of course, because that's why we're all here, right? Because we love the brain. So just briefly, this series follows best friends Pablo and Violet on these crazy out of this world adventures in a magical laboratory called the Maker Maze, where they're led on fun adventures, science adventures, by a kind of eccentric or kooky scientist who runs the Maker Maze called Dr. Crisp. And so where I'm going to start reading in Brain Trouble, they've already entered the Maker Maze and they're beginning level 1 of the science challenge. And they're accompanied by 2 twins, Skylar and Devin. So get ready, and here we go on a magnificent science adventure. Violet, Pablo, Skylar, and Devin couldn't believe their eyes. Floating in the middle of the room was a giant brain. It rotated slowly. Violet's mouth fell open as she moved closer. "I'd like you all to meet Bob the Brain," said Dr. Crisp. "He's so beautiful," said Skylar. "He looks like he should be in a museum." "Maybe a science museum," Violet laughed. Suddenly, a rumble echoed through the room. Bzzap! The rumbling stopped, and a hologram of the twins appeared. Skylar was painting a picture of her brother. Devin was standing in front of her, spinning a basketball on his finger. "Whoa," said Devin, covering his mouth. "How can we be here and there?" said Skylar. Pablo smiled. "They're just clones." "Yeah, the Maker Maze can make a copy of anything," added Violet. Bzzap! A hologram of Violet appeared. She was standing at a lab table holding 2 test tubes. One had a blue powder and the other held a yellow liquid. "Cool," said Violet. "I look like a real scientist." "You are a real scientist," Dr. Chris smiled. Bzzap! Then a hologram of Pablo popped into view. He was aboard a spaceship, sleeping in a bag attached to the wall. "I read about those. Astronauts have to sleep in bags like that so they don't float around and bump into things," Pablo explained. "Okay, makers, it's time to make a match. Maker Maze, activate the brain matching game," she shouted into her watch. Boom, snap, whiz, zap! A cloud of green smoke burst into the air. Crash! When all the smoke cleared, the makers saw Bob's cerebrum, cerebellum, and brainstem scattered on the floor. "As you can see," began Dr. Crisp, "the holograms are all doing something different." The makers nodded in agreement. "In each case, a different part of the brain is working extra hard to help them. In this level, you'll have to match the correct part of the brain with the correct hologram," said Dr. Crisp. "It doesn't look like Pablo's brain is working too hard," said Violet as she nudged her best friend. "Hey, space sleep is hard work," Pablo laughed. "And sleep is very important for your brain," said Dr. Chris, tapping her head. Dr. Chris cupped her hands around her mouth and yelled, "Ready, set, match!" The makers studied each hologram carefully. Hologram Skylar's arm lifted and lowered as she painted. Hologram Devin smiled as he gave the ball a swish to keep it spinning. Hologram Violet bit her lip and squinted her eyes through her safety glasses. She examined each test tube carefully. Pablo's hologram snored softly. "I think I'm using my cerebrum. I must be working on something important," Violet said. "Maybe I'm discovering a cure for the flu." "I bet I'm using my cerebellum. Look at all the different kinds of movements I'm doing," said Skylar. "Yeah, and my cerebellum is probably helping me balance the ball," added Devin. "I wish I could do that in real life." "Which means my brainstem is making sure my body keeps working while I sleep," said Pablo. "Okay, now we just have to move the pieces," said Violet. "How about," began Skylar. "We each move our own," Violet interrupted. "Well, I was thinking we could work together," said Skylar. "But we'll finish faster if we divide the work," Violet insisted. Pablo looked at his watch. We have plenty of time, he said, but maybe Violet's right. If we save time now, we'll have more time to finish the other challenges. Well, I think my sister is right, Devin said with his hand on his hips. We should work as a team. Violet huffed and tapped her foot. Why don't you two work together to move the cerebellum, she replied. I'll move the cerebrum and Pablo can move the brainstem. Devin rubbed the back of his head. If you say so, but the cerebrum is really big. You might need some help. Don't worry, said Violet. She flexed her arm. I'm tough. The twins shrugged and ran over to Bob's cerebellum. It was heavy, but together they were able to push it. Pablo picked up one end of the brainstem and decided to drag it. Violet ran over to the cerebrum. It was big. She walked around trying to figure out how to pick it up. Maybe I should ask for help, she thought. She bit her lip and checked to see what the others were doing. Everyone was busy moving their pieces. Violet decided she could do it alone. She brushed a few loose curls out of her face and got to work. Violet tried to lift Bob's massive cerebrum, but it was way too heavy. She grabbed it with both hands and tried pulling it. No luck. She tried pushing it. It scooted a couple of inches, but then Violet needed to take a break. Ring ding dong. Pablo had lugged the brainstem to the correct spot. Excellent. This challenge is off to a stimulating start, shouted Dr. Crisp. A few moments later, the twins pushed Bob's cerebellum into place. Ring ding dong. Yes, they cheered. The twins each gave each other 2 high fives and a fist bump. Violet dug her heels into the floor and pushed. She still had a way to go before the cerebrum was in place. Let us help you, said Pablo. I almost got it, Violet grunted. Pablo pointed to his watch. We'll be here all day if you try to push this big thing alone. Yeah, agreed the twins. But I'm so close, Violet whined. Not really, mumbled Skylar. Violet frowned. I'll show them, she thought. She pushed with all her might, but she had no strength left. Bob's cerebrum wouldn't budge. Violet stomped her feet in frustration. How come you guys got the lighter pieces? Pablo put a hand on his best friend's shoulder. Come on, Violet, this isn't a competition. Let's finish as a team. Violet sighed. Okay. The twins joined Pablo and Violet as they pushed the cerebrum in front of Violet's hologram. Ring ding dong. Dr. Crisp skipped over to the group. You rocked that level, makers. Dr. Crisp jumped in the air and played an air guitar. The twins turned toward Dr. Crisp. What's next? They asked, smiling. Dr. Crisp took the maker manual out of her backpack. The pages began turning. When they stopped, the page read, Level 2, Prism Goggles. Go through secret door number 3. She closed the golden book and tucked her rainbow hair behind her ears. Then she pressed the button on her watch. Out shot a purple laser. She used it to scan the floor of the room. Whoa, said the twins. What's she doing? Whispered Devin. Violet and Pablo shrugged. Beep, beep, beep. Dr. Crisp stopped in her tracks. Aha, she said. She drew an M on the floor with the laser. The secret door opened. Dr. Crisp jumped. Follow me, her voice echoed. The makers hurried over to the opening in the floor. It was pitch black. Violet sprang up in the air. Whee, she yelled. Devin's eyes grew wide. Is there another way, he asked, to get to level 2, finished Skylar. Pablo laughed and jumped. You get used to the weird things that happen in the maker maze, he said as he fell. Skylar grabbed Devin's hand and they leaped into the darkness together. So I hope you guys enjoyed that reading. Again, that was from book 2 in the Magnificent Makers series. And now, so a cool thing about these books is that in addition to having a lot of fun and science and adventure, they also have activities in the back that you can do at home. And so one of the activities is a brain bake. And so it's a little bit hard for me to show you how to do that virtually, but I do have a video that we're going to play right now that will show you how you can bake your own brain at home. Hello, everyone. My name is Theanne Griffith, and I am the author of the Science Adventure chapter book series, The Magnificent Makers. Today, I'm going to be showing you how you too can be a magnificent maker and bake your own brain. This activity is featured in the back of Book 2, Brain Trouble. Let's get started. The first step to baking your own brain is making the dough. And to do this, you just need 3 simple ingredients: water, salt, and flour. Next, you need to measure out 1 cup of the flour, 1/3 cup of water, and then 1/4 cup of salt. Then you'll add the flour and salt to a mixing bowl. Then we're going to slowly add the water and stir until the dough is no longer crumbly. If you make a mistake and add too much water, don't worry. Just add a little bit more flour until the texture is just right. Making mistakes is important for the learning process, and magnificent makers aren't afraid to make a few mistakes. Next, you want to sprinkle a little bit of flour on a clean surface like your countertop or a cutting board. Next, you're going to take your dough and roll it around in the flour until you can mold it easily. Now we have a nice piece of dough that's all ready to be shaped into a brain. The wonderful Reggie Brown, the Magnificent Makers series illustrator, has provided an outline that you can use as a guide when shaping your brain. You can make one big brain or divide the dough up into smaller pieces and make several smaller brains. It's up to you. Because I'm not such an awesome artist as Reggie, I decided to trace his outline using pencil on a piece of parchment paper. And voila! We're ready to bake our brains. If you want to make a bigger brain, it might look something like this. Now it's time to preheat the oven to 350 degrees. Make sure you have an adult help you. It's always important to make safely. Next, we're going to bake our brains in the oven for about 10 or 15 minutes. Remember, ask your parents for help. It's always important to make safely. While we're waiting, let's review some parts of the brain, which can be found in the Magnificent Makers number 2, Brain Trouble. The largest part of our brain is called the cerebrum. Our cerebrum is really important for things like learning and memory and decision making. Next, down below, we have the cerebellum. The cerebellum is really important for helping our body move and helping our muscles work together, and it's also important for balance. And then finally, we have the brainstem. It's a long, skinny part that sticks out the back. Our brainstem makes sure that we're always breathing, and it also makes sure our heart's beating. Once your brain is done baking, it's time to wait and let it cool down. Now that our brains have cooled, it's time to paint. And voila, we're done painting. I have the cerebrum in purple, the cerebellum in red, and the brainstem in orange. You can paint these brain structures whatever color you want, or you can just paint the whole brain whatever color you want. It's completely up to you. Let your inner creative, magnificent maker shine. That's it. I hope you had a lot of fun making with me today. I'd love to see what kind of brain bakes you guys come up with. Feel free to share anything you create using the hashtag #MagnificentMakers. And don't forget, you can find more activities like this in books 1 and 2. Okay, well, I hope you guys enjoyed that activity, but I didn't want to leave without doing a real live science experiment for you guys. So what I decided to do is an activity from book 3 in the series, which is called Riding Sound Waves. And this book is all about our senses, and in it, Pablo and Violet and their friend Henry make a model eardrum and figure out how our ears send signals to our brain so that we can hear. So what I'm going to do right now is do a little demonstration of how you too can make your own model eardrum. And it's so fun because it shows you that sound isn't just something invisible that's floating throughout the air, but as you'll see very soon, sound is actually something very physical. It's air molecules moving all around and bouncing all over each other. And all of those vibrations are passed onto your ear, and then your ear sends those signals to your brain. So if you want to see those sound waves at work, you can do this activity at home all by yourself, and you just need a few simple things. The first thing you need is a pot and a wooden spoon or something similar that you can make a lot of noise with, because this activity makes a lot of noise. Next, you need a bowl, and then you need some plastic wrap. Okay, and then the final thing you need is something that you're going to see move. And so in my case, I have sprinkles, but you could use rice, you could use, just about anything that's not too heavy. But I like sprinkles because they're colorful, as you'll see very soon. Okay, so I've already done the first step in this experiment, and that is putting on the plastic wrap. So that is it's really important that the plastic wrap is nice and tight. The first time I did this, I didn't make the plastic wrap tight enough and it didn't work. But that's okay, because magnificent makers aren't scared of making mistakes. And I just did it again and then it worked. So if you try it the first time and you don't see any movement, try making sure that plastic is nice and tight. Okay, so now that we have our model eardrum, I'm going to put on the sprinkles. And you just need a little bit. And actually what I'm going to do now, so you guys can see this, is put my computer screen down. Okay, and so I'm going to take a little spoonful. This is just about half a teaspoon. And I'm going to try and put it in the center like that. You guys see that? You really don't need too much at all. Okay. Now, here comes the fun part. I will give you guys a warning. I'm going to shortly make a lot of noise. So if you're sensitive to noise, or maybe if you have earphones in, you might want to turn down the volume because it's going to get pretty loud here in a second. So what I'm going to do is I'm going to take my pot and I'm going to take my wooden spoon and I'm going to beat on it right over my eardrum, and you guys are going to see what happens. Okay, here we go. Okay, it's gonna get noisy. In 3, 2, 1, let's ride some sound waves. Did you guys see that? Do you guys see the sprinkles all over my desk now? Let's see, we can do that one more time. Let's add a little bit more sprinkles. That's so fun. I can never just do it once. Oh, I love making a mess. Okay. In 3, 2, 1. Woo! Those are some serious sound waves. So I hope that didn't hurt anybody's ears, because like I said, it gets pretty loud. But as you can see, that's exactly how our eardrum detects sound. It picks up the vibrations, and those vibrations are sent via nerves to our brain, and our brain can learn to make sense of all of those sounds. And we begin to recognize sounds the more and more we hear them. So that's the end of my live demonstration. I hope you you guys enjoyed that. And again, I'm the author of The Magnificent Makers. There are currently 3 books out in the series, so I hope you go and pick those up, and I'm happy to answer any questions you might have.
Oh my God, we loved that. That was fantastic. And we like making messes too. So this is great.
I have a little bit of a mess to clean up, but that's fine.
Thank you so much. So we have a number of questions here. One is, what inspired you to write children's books? I have never heard of a neuroscientist children's book author.
Yeah, so I've always had 2 kind of main passions. I've always been really passionate about science, and I've always been really passionate about storytelling, reading and writing. When I was a little kid, I was really fascinated by how the world worked around me, and I was, the kid like playing in the dirt and finding bugs and trying to capture lightning bugs and picking up tadpoles from the creek. I just always have that passion for science. But as I said, I always had this passion for storytelling, and I would go to the library and check out big stacks of books, and Magic School Bus was on the top of my pile most days, and so I guess I had these 2 passions, but for whatever reason I ended up sticking with science for most of it. But I always had this desire to continue to, I wanted to write books, and I didn't know exactly how and when that would fit into my life, but I knew I wanted to incorporate it somehow. And then in about 4 years ago, while I was on maternity leave with my daughter, I said, let's just go ahead and do this. I'm reading her all of these books, and I'm kind of feeling that dream that I had put aside for a while emerge in me. And I said, let's go do this. And I wanted to write about science because I really care that kids get interested and engaged in science and find science cool and fun and feel seen in the scientific community. So those are a bunch of reasons why I was motivated to become a children's book author.
That's awesome. Thank you. There's another question here. Sleep is very important to your brain, as your book explains. Do you think kids who get inspired by stories like yours might become better advocates, actually, for their own need for sleep, given all the challenges that many kids face?
That's an interesting question. I would really hope so. A lot of another kind of almost linking the first question and this question, a big motivating factor for me in reading these books is having kids feel seen, having kids identify with the characters, So if they're engaging with the books and feeling like, oh, Pablo knows that he needs to sleep, so his brain works better. Pablo's kind of like me. I should sleep too, and I should make sure I get enough sleep. That would be like amazing if I was making those kind of connections in kids' lives. Like I would feel like, my job is done here.
That is awesome. So here's another one is that we love that magnificent makers aren't afraid of making mistakes. It is how we learn. Do you think our current educational system does enough to encourage kids to explore on their own, experiment, take risks, and make mistakes?
That's a tough one. I think teachers are working really hard to make sure that kids understand that, perfection is impossible and that it's okay, to fail. But I think that the way our educational system is designed, right, we have these standardized tests that make kids feel like, oh, I have to get a certain score, otherwise maybe I can't go to the next grade, or maybe I'm not smart enough, So my kids are not school aged yet. So I actually don't have I'm not like in the weeds of school. So it's hard for me to say whether or not, across the board, we're encouraging failure enough. I know that when I was a child, they were not. It's hard, that was a while ago. So I would hope that things have gotten better. But I see it all the time, actually, in the lab. You have people who come into the lab who are very smart and can, memorize a bunch of things. And then they realize that, going through experiments, they're making mistakes, and they feel so bad and they get so down on themselves about it, And so what that's telling me is that they've been trained not to make those mistakes. But science is literally like 80%, I wouldn't say necessarily 80% mistakes, but 80% of the time something doesn't work, right? It's that 20% that keeps us going. So I don't know whether my instinct is to say that we can definitely do better. on the accepting failure front, and encouraging exploration, encouraging, what I call creative thinking and asking questions. And it doesn't matter if, the question's already been answered, but just kind of letting kids the freedom to explore their inner scientist, allowing them that space that without instilling any insecurities.
That's wonderful. We have a question about, actually, an interesting question about, they say, that was great. What is the role of metaphor for you in describing and understanding actually complex neuroscience concepts?
Yeah, I always try and so when I'm trying to break down some of these concepts for kids, I actually pay a lot of attention to some of the logic that my daughters use. And they are much smaller. They're only 3 and 2. So they're much smaller than the kids that I'm writing for. But I start to kind of figure out what connections they make to things, right? And that really helps me figure out how to relate these really challenging, right, scientific concepts. Like, we're talking about cerebrum and cerebellum, right? Like, those words are hard. In the actual text, we decided to spell them phonetically so that we can help kids out and not have them feel intimidated by all of these kind of crazy words, right? And so when I'm trying to write the books, I really try and pick up on what they make connections with. And I try to use those, for the metaphors in the book. And I also just try and use simplistic language without losing the meaning, right? While still maintaining meaning.
Yeah, no, I mean, you explained, like, sound waves and what's going on, like, very, very clearly, and it was accurate. So, so I am curious what you recommend aspiring neuro volunteers to do to inspire younger folks to be more neuro-minded, is what they said.
Yeah. First of all, having like Noggin Fest style events is a great way to start. I've been participating in brain fairs since I was a graduate student. I got immediately, into outreach and whatnot. And I think that kind of FaceTime between scientists and the community is so important. And especially as it pertains to neuroscience, right? I think that neuroscience is it's not a new field by any stretch of the imagination, right? We've been studying the brain since we've had brains, but compared, in terms of it being an official major and an official degree and all of these things, I don't think it's on any core curriculums for elementary school, for example, which I personally think it should be.
We actually went through like the federal science standards and NGSS, and they mentioned the brain I think it was twice in the entire K-12 curriculum.
Exactly. So, I think that there's a little bit of a hole there. And so hopefully having this interaction, that's why I do so many of these events. That's why I love doing outreach events, is because I want FaceTime with the public. I want them to see what a neuroscientist looks like. And hopefully, it inspires them to feel like, oh, she's a neuroscientist. I could do that too, that's what I can do.
Very inspiring. We have one last question I think we have time for is, when do you remember first really becoming interested in science specifically as a child? What topic first sparked your interest?
I've always loved like animals. I think it's a very typical thing, right, of kids. Kids who like science, or at least the biological sciences. So I was really into animals. I wanted to be a veterinarian for a long time. And then somehow that shifted to doctor. And then it was actually, this isn't quite the question, but in high school, I took an AP bio class and was introduced to neuroscience there. And I haven't looked back since. So I always had this interest in science pretty much since I can remember. Like, I don't remember the aha moment. I just remember always loving science and being able to think about science in my head kind of easily. Like, I enjoyed thinking about it, and like kind of making up little science worlds like I still do today. Clearly nothing has really changed. I'm still making up science stories.
Grant funding for that now. Actually,
so I would say, like, I've been a scientist forever, and then I was specifically sparked by neuroscience when I was about 16 in AP Bio.
Yeah, Dr. Griffith, thank you very, very much. We really appreciate you joining us, and that was a wonderful experiment in reading from the book. So, cheers.
Thank you so much for the invitation. Have a great weekend, guys.
Yeah, thank you, Theanne. I really appreciate your work that you're doing and how you're making neuroscience even more accessible for everyone, including children. So that's super awesome. And for our final speaker of the evening, everyone, I would like to welcome Dr. Chris Stoffer, who has an extensive background in psychopharmacology, psychotherapy, and psychedelics. So sit tight for just a moment while we get him set up, or if he's already ready, he'll just pop on over.
Hello. Um, Yay, Noggin Fest. This is delightful. Let me see if I can share my screen. All right, so I am going to talk about psilocybin therapy. We are in Oregon, kind of at the center of the psychedelic renaissance with some legislation that passed recently. I'll talk a little bit more about later. But I'm going to focus on psilocybin and specifically psilocybin therapy. So what is psilocybin? So there are over 200 species of psilocybin containing mushrooms. And what sets psilocybin mushrooms apart from other mushrooms? I think this does a good job explaining it. So you may have heard psilocybin mushrooms being referred to as magic mushrooms. So here you can see, I don't know, like a portobello on the top left saying, I'm tasty and nutritious. And then any amateur mycologist will know to avoid mushrooms with red on the cap or the stem. And this, we'll call it an Amanita muscaria, says, I'm poisonous, careful now. And then we have our psilocybin mushroom declaring, I unravel the very fabric of reality. So how does that happen? Psilocybin is an alkaloid, which is a class of nitrogen-containing compounds produced by a variety of organisms, microorganisms, plants, other animals, and they typically have some sort of pharmacologic effect. And once psilocybin is ingested, the phosphate group is removed, and that it actually turns into psilocin, which is the active drug that acts on our receptors. And for comparison here is serotonin, one of our body's major neuromodulators. And psilocin actually binds to the serotonin 2A subtype receptors, and that's what we believe to be the primary mechanism for its psychoactive effects. And a little bit of interesting trivia. When psilocybin mushrooms are picked or damaged, they get this characteristic blue discoloration. And that's triggered, so when there's cell damage, it also triggers this dephosphorylation. And the psilocin molecules are less stable, and so they join into polymers. And they look very similar to indigo dye, which is used to make blue jeans blue. So this is a really cool visual representation of functional brain networks, or parts of the brain that activate together. And this is a brain in resting state with a research participant who was administered placebo compared to a research participant administered psilocybin. So there's a lot more going on, a lot more crosstalk between different parts of the brain that maybe don't usually communicate. There's also evidence that psilocybin increases neural plasticity, which is the ability of neural networks to change through growth and reorganization. So you can see that might facilitate this process. So before we knew anything about the neuroscience of psilocybin mushrooms were consumed as part of spiritual ceremonies, likely for thousands of years. And the Aztecs called them the flesh of the gods. And then with Spanish colonization of Mesoamerica in the 16th century, we got the first written accounts of psilocybin mushroom use. And then this, unfortunately, was also one of the early wars on drugs in which Catholicism condemned the ritual use of psilocybin mushrooms and essentially pushed these practices underground. And it wasn't until 1953, which is about 400 years later, when Maria Sabina, Mazatec curandera, or a Latin American traditional healer, allowed Westerners to participate in a healing ritual using psilocybe mexicana mushrooms. And in this picture, you see Maria Sabina giving a mushroom to Robert Gordon Wasson. He was an amateur mycologist, and he was one of these Westerners. And he brought some of these mushrooms back to the US and introduced their psychedelic effects to a wider Western audience for the first time in 1957 via this Life magazine cover story, The Discovery of Mushrooms That Cause Strange Visions. And just to recognize, so Maria Sabina actually later regretted having introduced these spiritual practices to Westerners. After this Life magazine article came out, her village attracted a lot of psychedelic tourism, which really upset the culture. And she ended up dying in poverty at the age of 91 in 1985. But so when these mushrooms were introduced into the West, Gordon Wasson sent samples of them to Albert Hofmann, who is the Swiss chemist who first discovered the psychoactive effects of LSD. And Albert Hofmann isolated the active component, psilocybin, and he worked for a company called Sandoz Pharmaceuticals. And Sandoz Pharmaceuticals manufactured and distributed pure psilocybin tablets around the world for research purposes under the brand name Indocybin. And then by 1969, so we had the Summer of Love and widespread use of psilocybin, LSD outside of a medical setting. And in 1970, as part of Nixon's war on drugs, the DEA made psilocybin a Schedule I substance through the Controlled Substances Act. And Schedule I substance, that's the most restricted category of controlled substances. And it usually, the criteria for it is that there are no medical uses, it's not safe to use even in a medical context, and can only be used in highly regulated research. But we come to find out in more recent years that, we didn't really have any reason to think that psilocybin was unsafe. It wasn't really made into a Schedule I substance because it had no potential medical use. There were studies already out showing that it did have potential therapeutic benefits, but because it was associated with the anti-war movement, it was put into this more restricted category. And this prohibition essentially stopped legal research and led to the closure of psychedelic research programs around the world. And this is a chart showing the number per year of new scientific peer-reviewed articles on psilocybin. You can see the introduction of psilocybin into the scientific lexicon in the 60s, a little bit in the 70s, and then there was a dormant period for several decades. But in recent years, the topic of psilocybin in the scientific literature has increased exponentially. And psilocybin therapy, which I'll talk about now, has been actually given breakthrough therapy status for the treatment of depression by the FDA. And breakthrough therapy status means that the FDA believes, based on existing evidence, that psilocybin therapy offers an advantage over existing treatments for depression. And breakthrough therapy designation essentially expedites a new medication through the FDA drug development process. So that means that psilocybin therapy is on its way to becoming an FDA-approved treatment. So what is psilocybin therapy? So the current model, which has been refined over decades of research, uses the key ingredients of drug, setting, and support. And so the therapy you can see here is typically administered in co-therapy teams. And set and setting is a term that really highlights the importance of the set or the mindset of the participant and the facilitators, including mentally preparing for the session, having an intention going into it, and then the physical setting or the therapeutic environment is set up to be comfortable, eliminate any distractions. And we found through research that set and setting can greatly affect the subjective experience while on psilocybin. And the model that we use, so usually we will try to avoid focus on the external world, and participants are offered eye shades, headphones with a pre-selected playlist, usually music without vocals, or at least without lyrics. And they're instructed to direct their focus inward. And a motto that came out of the research in the 60s was trust, let go, be open. And the facilitators are there to assure them that even if frightening or upsetting or challenging material arises, the facilitators are there to keep them safe. And we invite them to move into whatever challenging experience comes up for them, as a way of potentially learning about maybe a rut that they're in. We also loop in social support in whatever way we can. So family, partners, other social support network that can help with the integration process afterward. So this is a typical protocol for psilocybin therapy. The number of sessions can vary depending on the study or the setting, but preparatory sessions are typically used to set intentions, provide psychoeducation around how to therapeutically maximize the effects of the psilocybin dosing sessions. And then the dosing sessions, typically if there's more than one, they're spaced several weeks apart, and they usually are started in the morning and go for about 6 to 8 hours. And then each dosing session is followed by a series of integration therapy sessions, which gives the participant an opportunity to work with a therapist around what came up during the dosing sessions. Usually material comes in symbolism. It's like material from the unconscious. And so it's very right brain sort of visions or metaphors. And so it's helpful to sort of go through what came up during the session and make sense of any insights, and then help implement these insights into their ordinary lives and ordinary states of consciousness. So this is a recent study from Johns Hopkins Center for Psychedelic and Consciousness Research. And this shows the effects of psilocybin therapy for depression. So participants in the study had depression on average for 21.5 years. And their current depressive episode was over 2 years. And they were randomized to either receive psilocybin therapy, which is the orange squares, or to get put on a wait list, which are the blue circles. And you can see that compared to the wait list, those who got psilocybin therapy right away had a significant reduction in their depression symptoms at 5 and 8 weeks post-treatment. This is also a study from Johns Hopkins. This protocol was a little more rigorous, a little bit more visits, and it was for smoking cessation. So the participants in this study had smoked on average 1 pack a day for 31 years, with an average of 6 lifetime quit attempts. And when you looked 6 months out after psilocybin therapy, 80% remained abstinent and were no longer smoking. And then after a year, that dropped off to 2/3. But this is still a lot more people than the traditional treatments that we have for smoking cessation. And then the last one I'm going to show you is a study showing psilocybin therapy's effects on reducing drinking days for individuals with alcohol use disorder. These individuals had alcohol use disorder for an average of 15 years. And you can see where the green arrow is pointing. That's where they had their first psilocybin treatment. And there was a big drop off in the percent of drinking days and the percent of heavy drinking days that lasted out to the final outcome measure at 36 weeks. So many of you may be familiar with some legislation that passed in Oregon this past year. Measure 110 decriminalized personal non-commercial possession of a small amount of any controlled substance, which is a really important step toward repairing some of the harms from the war on drugs. And this includes personal possession of up to 12 grams of psilocybin mushrooms. And then Measure 109, the Psilocybin Mushroom Services Program Initiative, authorized the Oregon Health Authority, after a 2-year development phase, which is about to start this month, to create a program of licensed service centers which will administer psilocybin mushrooms on site in the presence of trained facilitators. So it's not like medical marijuana where you would take the psilocybin away and take it home. It's something that you would get administered and then have a supervised session with trained facilitators. And a psychiatric diagnosis will not be required. This does circumvent the FDA drug development process that I was referring to earlier, which is researching synthetic psilocybin. I think I had a picture of a little capsule for patented medical use. Whereas this Oregon initiative will allow any Oregonian over 21 years old to have access to a facilitated session using natural psilocybin mushrooms, which is pretty groundbreaking. I don't think there's been a lot of research on psilocybin mushroom use. And here I just want to point out a couple of resources. So the Zendo project provides psychedelic peer support, and they have some great resources online. And when we get back to gathering in large groups, you can usually find a Zendo harm reduction tent at music festivals where there are trained facilitators who provide a supportive set and setting and can help turn a potentially bad psychedelic experience into something meaningful. And then on April 14th, the Psychedelic Peer Support Line goes live, and this will provide free, confidential, 24/7 peer support by phone or text message. And it's 62-FIRESIDE or 623-473-7433. And then lastly, just a plug for my lab at OHSU and the Portland VA Medical Center, the Social Neuroscience and Psychotherapy Lab. And we're planning a psilocybin-assisted therapy clinical trial for veterans with methamphetamine use disorder. We have a bunch of other exciting projects in the pipeline. Also, OHSU has a psychedelic medicine and research collaborative with over 70 faculty and trainees. There's a new OHSU medical student psychedelic science interest group, which I'm sure would be more than happy to collaborate with other university organizations in Portland who may have similar interest groups. And my website and Twitter are at the bottom. And I'll end there and see what questions we have.
Thank you very, very much. This is extremely topical, and there's a lot of interest, and there's a lot of interest in terms of the questions asked tonight. The number one question, I think, is how do you get involved in being a psychedelic therapist? How do you become a trained facilitator? Is there a school or a training certification that's involved?
That's a really good question. A metaphor that we use is that we're kind of flying the plane as we're building it. There are professional organizations. I think the biggest one right now is the California Institute of Integral Studies. They have a big program. I think this is their 4th year, and they've gone from like 20 students to I think 300 this year. You have to be a licensed therapist to get into that program. But for Oregon, for the Oregon Psilocybin Services Initiative, it's written into the legislation that people don't have to have a higher education. They don't have to have a therapist degree or a doctoral degree. They just have to get certified through whatever the Psilocybin Advisory Board and the Oregon Health Authority decides is proper certification. And so I think the sort of leading contender right now is the School of Consciousness Medicine that's launching. I think you can go online and fill out an application to get trained through them. And I believe they're sort of the front runner for being the psilocybin services certification program here in Oregon.
So they just appointed that board, is that correct? The members of that board.
That's right.
Yeah. So this is just the very beginning of this process. So how has Measure 109 affected psychedelic research in Portland? Has it had an impact on research programs?
Well, there hasn't been any like clinical trials in Portland yet. And I just moved up here from the Bay Area last year, where I was doing psilocybin research and research with MDMA. And so I moved up here with the intent of starting a psychedelic research program at OHSU. And then this measure happened, and I think things opened up. There's suddenly a lot more activity, a lot more interest. So it's exciting, but there is that interesting tension between psilocybin mushroom therapy, with certification where you don't need a higher degree, and mushrooms are dirt cheap, and they're I think looking to provide access to a wider range of people, whereas the medical model and the sort of typical big pharma drug development model, I think would actually limit access to this treatment for people. So I think it's an active war right now.
I'll bet, because sometimes you only need to take it once, apparently, right? So
yeah.
As opposed to being hooked on something. So what happens in the brain? This is a big question. What happens in the brain, with psilocybin and other psychedelic therapy that relieves depression?
Yeah, it's an open question. I think there's a lot of mystery. I think the easiest answer is we don't really know. And I showed you the brain map. We know what receptors psilocybin hits, but each person's process when they go through a psilocybin session is so different. And I think, the ruts that people get stuck in are so individualized. And we really do have this, trust, let go, be open motto. And we encourage people to stay with whatever comes up. And it's such a wide range of things that come up for people. Sometimes it's very spiritual and joyous, or, happy. Sometimes it's terrifying or sad, or there's a lot of grief. So it's kind of, we trust that whatever's coming up for people is what they need to work through. And we just try to help them stay present with whatever that is.
So we have another question here. Do you know of any studies that followed users years after treatment, or are we not there yet?
I don't think we're there for psilocybin. Well, actually, that's not true. I think the tobacco use disorder study, well, that was a year. There's a year follow-up. I'm sure there's something out there, but I'm more familiar with MDMA therapy, which is another sort of form that we're using for PTSD, and they have studies that are like 3+ year follow-out, and people tend to do better. And I'm assuming that something like that happens with psilocybin, where they're, even with one session or two sessions, their process continues to unfold for weeks and months and even years. Sometimes they continue to sort of recognize insights and integrate insights into their life. And I think it's almost like a crash course in teaching depth meditation to someone. And, even though they just have that one session, they take those tools and apply them in their life afterwards.
That's really interesting. So one last question here we have is, Oregon just legalized psychedelic therapy and announced an oversight panel. What are your thoughts on who was just appointed to that panel?
I know several other people on the panel, and I think it's a great group of people, and I'm looking forward to seeing what their recommendations are. At the end of the 2 years, they'll sort of give their recommendations for what the certification program looks like, what it looks like to be a certified mushroom grower, all these things. And then the OHA decides which of those recommendations to take, or how to roll this program out eventually.
That's good. That's very promising. So thank you very, very much for joining us at Noggin Fest. We really appreciate this.
Of course. Yeah. Thank you.
Exactly. Cheers.
Yes, thank you, Dr. Stauffer. Next, we are going to unwind just a little bit and listen to Portland's own classical guitarist, Ryan Walsh. But you'll want to stay tuned afterwards for the announcement of our raffle winners. And after that, we will wrap up Noggin Fest for the evening. So sit tight and enjoy some beautiful guitar music from Ryan.
I hope that everybody can hear that okay. And I had a moment of panic there that I forgot to unmute myself, so I hope that sounded good. Good. So that first piece is called Spanish Romance, and it's a really old folk song from Spain that doesn't have a composer. Yeah, I'm gonna play two more pieces here because we're keeping it short and sweet tonight. And they're going to both be original. This next one is called Zephyrus, named after the Greek god of the west wind and spring. And this feels like an appropriate time of year to play that. So it's just a really short little piece. All right, and I'm gonna play one more original piece here. This one's called February. It's about the month that we just had. And I actually wrote it during the first snowfall that we had while we were all stuck inside for a few days. And yeah, it's another short little prelude. And I just want to say that if you want to listen to any of this music, you can go to my YouTube channel. I think it's being written somewhere on screen, but it's Ryan Walsh Guitar is the YouTube channel. So, okay, here's another piece that I wrote last month. All right. that's what I got for you today. Thank you. And it's been an awesome event. So thank you for letting me be part of it.
Yeah, thank you, Ryan. That was really beautiful performance. I always really like hearing classical guitar. I feel like it's very calming and soothing and emotional. And I just want to thank everyone else once again for joining us this evening. We were so excited to bring our love of neuroscience right into your homes, and we really appreciate all of you who stuck around and uh, wanted to see everything that we had to offer tonight. So thank you very much. If you enjoyed this event and you want to give back a little, Northwest Noggin always deeply appreciates your donations. We'll put a link in the chat if you'd like to make a donation so that this kind of outreach can continue, and so we can continue to inspire our community for years to come. And hopefully, here soon enough, we'll be able to do it in an in-person format rather than having to be so far apart from one another. So now, to wrap up the evening, one of our awesome volunteers, Abigail, or she likes to be called Abby, she just told me, is going to hop on and announce our 4 raffle winners of the evening. Go ahead, Abigail. Abby.
Hi, all. So the first winner is Carol Newman. The second winner is Jennifer Teachworth. Our third winner is Peter Noble. And finally, our fourth winner is Krista Schramm. So congratulations to all of you for winning. And we encourage you to pick up your prizes in person. But if that's not feasible, we can negotiate shipping options with you guys. And I will be emailing each of you with details to the email address you provided when you purchased the tickets. Thank you.
Yes, thank you, Abby. And just to wrap it up a little bit on what everybody got, our first prize is going to be our Paxton Gate donation package, along with a brain cross stitch and a Northwest Noggin Fest sticker. Our second winner is going to receive this lovely pipe cleaner neuron that's hanging behind me, as well as a brain cross stitch pattern and a Noggin Fest sticker. And then our third place winner is going to get one of these lovely 3D printed brains, as well as a cross stitch brain and a Noggin Fest sticker. And then our fourth winner is going to get our little pipe cleaner astrocyte and a really cute little knit neuron doll that one of our volunteers hand knit for us, and then as well as a Noggin Fest sticker as well. So thank you, everybody, and thanks, Abby, for your help with the raffle moderating, and congratulations to all of our raffle winners. That is going to conclude our Noggin Fest 2021. Thanks again so much to all of our dedicated Northwest Noggin volunteers who made this night possible. Thank you to all of our speakers and musicians who participated this evening. And of course, thank you very much to our audience for coming out and sharing it with us in our love of science, neuroscience, music, and art. We hope you learned a lot and had a good time. And we look forward to seeing you all next year at Noggin Fest. Again, hopefully in person next time. So take care, everybody, and have a really good night. Thank you very much.